The zebrafish is a staple inhabitant of freshwater aquariums, the size of a goldfish. It's also the subject of research by thousands of scientists worldwide. Its embryos are large and transparent, making it handy for studying vertebrate development. But how did this particular fish come to be so widely studied? To understand this, we have to go back to the Manhattan Project. Aaron Novick was a chemist who worked on plutonium production for the first nuclear weapons, but seeing the devastation of Hiroshima and Nagasaki caused Novick to become a pacifist and anti-nuclear campaigner, and also led him to move into biology, describing his change of fields as a move from death to life. Novick established the Institute of Molecular Biology at Eugene in Oregon in 1959. Shortly afterwards, he hired a bacteriophage researcher called George Streisinger. Bacteriophages, viruses that infect bacteria, had been used as a model system to establish many of the basic principles of molecular biology, such as the nature of mutation and of genes, and the importance of DNA. Streisinger had worked on phage with a number of big names in early molecular biology, including Salvador Luria, Max Delbruck, and Seymour Benzer. Streisinger was keen to develop a model system that could be used like phage to investigate vertebrate biology. He was a keen naturalist and tropical fish enthusiast, and so he knew that zebrafish were easy to keep as pets. However, establishing techniques for raising zebrafish on a mass scale and developing methods for analysing them was a long-term undertaking. Then, as now, science funding was based around a model of short-term research projects, and this kind of long-term infrastructure development was hard to get money for. Fortunately, Streisinger's boss, Aaron Novick, perhaps inspired by his father's socialist beliefs, operated a regime at Oregon where funds were redistributed between projects, and where there wasn't too much pressure to produce papers. Colleagues who were turning out publications and getting grants thus subsidized Streisinger, who published nothing on zebrafish during the 1970s. Streisinger's lack of publications was partly due to the fact that he was busy as a political activist, protesting against nuclear weapons, the Vietnam War, and damage to the environment. However, Streisinger's lack of papers was also due to the lengthy process of developing techniques that would allow the kinds of mutation experiment he had carried out on bacteriophages to be carried out in zebrafish, with their vastly more complex genome. He developed methods for carrying out mutagenesis and genetic mapping in fish. He also grappled with the problem that, like most vertebrates, zebrafish are diploid organisms, with 2 copies of each chromosome and thus of each gene. This makes it hard to carry out genetic investigations, so Streisinger developed methods for creating haploid embryos with 1 copy of each chromosome and for creating double haploid fish that are homozygous at all locations. Using these double haploid techniques, he went on to create clone lines of genetically identical fish, and this finally got him a publication, making the front cover of Nature in 1981. Tragically, just as zebrafish research was taking off in the early 1980s, George Streisinger died of a heart attack whilst taking a scuba diving exam. But this was not the end for zebrafish research in Oregon. Streisinger's colleagues continued his work. Chuck Kimmel carried out work on tracing the fate map of cells and tissues in the developing zebrafish embryo. Monty Westerfield wrote a primer on methods for rearing and studying zebrafish, and today he heads up ZedFin, the database of all things zebrafish. This zebrafish work in the 1980s helped reveal the remarkably high degree of evolutionary conservation of vertebrate development at the molecular level. The fact that many aspects of development remain the same between fish and humans makes the zebrafish a useful model for disease research. In the 1990s, a new centre of zebrafish research developed in Tübingen in Germany, when renowned fruit fly geneticist Christiane Nusslein-Volhard decided to turn her hand to fish. She had previously carried out large-scale mutation of Drosophila fruit flies to discover genes involved in development, and she now applied this approach to zebrafish. Between 1993 and 1995, a big screen for zebrafish mutants was carried out by her Tübingen lab, and by a lab in Boston headed by her student Wolfgang Drever. This produced 4,000 different types of zebrafish with lethal mutations that prevented their normal development. This collection of mutants provided ample avenues for further research into the genetics of vertebrate development, and as a result, the number of research groups working with zebrafish increased enormously. Today, the zebrafish has had its genome sequenced to a high standard, and an increasing array of tools are available for carrying out targeted genome modifications. Thus, the fish that George Streisinger turned into a research tool is more important than ever to science. This talk is largely based on Jim Endersby's excellent book, A Guinea Pig's History of Biology, which describes the development of modern biology by means of telling the stories of a number of model organisms. This was supplemented by Grunwald and Eisen's account of zebrafish history in Nature Reviews Genetics.